Hello, and welcome back to Beyond Networks, the evolution of living systems. This is sort of part two of last lecture, which was part one. So let me start this by sort of giving you a very sort of conceptual summary of what we did last time. So we looked at developmental system drift, in particular compensatory system drift, in these different fly species, where the gap domains, the gap gene system, was able to compensate for very big variation, absence of presence of different maternal gradients, through a tuning of the different interaction strength between the gap genes. And so what happened is that these different interactions shifted different nuclei in the embryo to different dynamical regimes in configuration space, somewhere in between phenotype space and genotype space. So the mechanisms that we found for this compensatory drift lay in switching the behavior, from shifting to stable domains. So from sort of this damped oscillator regime to switching regime that we encountered much earlier. We're going to revisit the gap genes in a bit, but what's really important here is that what's determining the evolvability of a system is the geometry of configuration space. So the explanations that we need lay somewhere in the middle of this genotype-phenotype map. And we sort of did, in this last lecture, we connected the changes in these dynamical regimes to the expression outcome. And today we want to sort of connect them back to the individual interactions, the mechanism, the network underneath. Okay, but we're still sort of busy with the structure of this genotype-phenotype space and evolvability, which, as we've also seen last time, requires a bunch of different conditions. So we looked at canalization, robustness the last time. We didn't really look at developmental constraints, but we've talked about them a bunch of times in the course already. So today we're going to focus on these sort of factors up there, which are, these conditions are flip sides of the same coin. So the covariance of related traits means that a lot of traits are not independent, and the modularity of the genotype-phenotype map means they're sort of independent at the same time. And so this sort of state of being quasi-independent is captured, of course, very well in this quote by Herbert Simon that I've shown you a couple of times already about the world being near decomposable. So everything is connected, but some things are more connected than others. The world is a large matrix of interactions in which most of the entries are very close to zero, and in which, by ordering those entries according to their orders of magnitude, a distinct hierarchic structure can be discerned. Now that we've discussed WIMSAT so much, we also know that in this biopsychological thicket that we're in biology, that these levels are not neatly separated. So these different levels of organization can be quite complicated to discern, and also that there are always many valid ways to decompose a system. So we're going to look at different ways of doing that. We've done that before, but we're going to sort of revisit this here. And so the basic problem that we're looking at is modularity today. Modularity is reflected in the existence of trait. Why are there discrete character traits at all? So phenotypes are modular, and we can observe that. Okay, so that's just a fact of life. What you see here are different tetrapod limbs, vertebrate limbs that are adapted to different sort of activities, functions, from the whale fin to bat and bird wings. And you can see this beautiful sort of variations on a theme, right? And these variations on a theme can only occur if selection can work on this trait in a quasi-independent manner from the rest of the body. If you would have every mutation being completely pleiotropic, so you would affect everything all the time, you couldn't evolve. It's quite obvious to see that. And so character trait modularity is a prerequisite for evolvability by limiting pleiotropic effects. And that's very important. Okay, so without modularity, no evolution. We also know that these traits, these character traits, I hope I convinced you by now, are generated by metabolic, physiological, and developmental processes, which constitute a mapping from genotype to phenotype. Also nothing new. So from that, it follows logically that if you have discrete modular traits, as shown here, the Lebowski and a bunch of his, different limbs he could have, it follows logically that you must have a modular epigenotype, in the words of Waddington. Okay, so the structure of this genotype-phenotype map must be modular as well. So the question is not, does this modularity exist? The question is, how do we find it? How do we best characterize it? And that is the central problem here. So this problem has been recognized a long time ago. Here is Joseph Needham in 1933 writing that generative processes are separable from each other to some extent. But again, the individuation of processes is a difficult problem, remember, so it's really much harder to draw boundaries around processes than things. And so there are some real issues here that we need to take serious. So let's look at some sort of different ways in which we can characterize this dissociability of generative processes. But before we can do that, we need to sort of think quickly, what is modularity in the first place? What are we looking for in the first place? Let me give you a definition that's by Lenny Moss here in the Cycles of Contingency book. And he writes, a module is a unit that is a component part of a larger system. And yet, and this is important, so the first part, not controversial, everybody knows that. The second part, we often forget a little bit. And yet possessed of its own structural and or functional identity. In order to be a module, you need to be or do something different than the rest of the system. Okay? A module can be any biological entity, a structure, a process, a pathway, or a collective of entities even, with more internal than external cohesion. Cohesion is an important term. What do we mean by that? Modules can be delineated from their surroundings, and their behavior emerges as a result of the integrated activity of their parts. That's sort of saying the same thing in different words. And that's very important. It may be a little trivial, but it's important. Modules must be extended in time. They must persist long enough to exert their effects. Otherwise, they're not really interesting. But their stability is something that needs explanation. And of course, last but not least, very important, modularity is always a matter of degree. Okay, so it's never 100% because no part of a complex system is independent of the other parts completely. So the classical way, of course, to characterize modularity is genetics. And it's very simple. You do a mutagenesis screen, just as in the case of the segmentation genes of Drosophila. And not only were Nusslein, Volhard, and Wieschaus able to identify all the mutations that affect segmentation, but they were able to group them into different categories that resulted in different phenotypes. For example, on the left here, you have two mutations, gooseberry and patch, that belong to the segment polarity category. They affect every segment, and in particular, the tissue polarity within every segment. Okay, so they're all similar. They're all different, but they're all similar in that particular way. Then, to everybody's surprise, there's the category of the parallel genes, where every other segment is missing. Could be the odd or the even ones, but that's what's characteristic. And then the gap genes, of course, my favorites, where a whole body part is missing. And it turned out when people did sort of the fine-grained genetic analysis, of course, of these segmentation genes, that they were also grouped together in these hierarchical tiers of the segmentation gene network. So the phenotypic sort of consequence reflected clearly something about the underlying processes here. So genetics, very powerful way of identifying functional modularity, okay? But there are some limitations, and we've been over all of these before. So I'm gonna go very quick here. So one, of course, is that any kind of perturbation-based functional approach will only give you a list of parts, sort of the necessary but not sufficient types of explanation. So you don't know how everything works together again. We've only done the decomposition, not the recomposition. There's a problem with correspondence because modularity is not perfect. So you always have a bit of pleiotropy, and especially in developmental biology, it's very bad because genes, especially developmental regulators and signaling factors, often contribute to more than one character trait. So one gene, many traits, but also one trait, many different mechanisms. Homologous traits are often generated by different network mechanisms because of developmental system drift. We've talked about that last time. And okay, I'm preaching to the converted, hopefully, here by now. These sort of explanations are never really process explanations. They're always thing-based, substance-based, momentary explanation, and can't explain the diachronicity, the temporal sort of extension of what's going on. So networks based on genetic and molecular decomposition, they are great. Okay, that's our door, foot in the door. Very rough, but it only is a very rough first sketch of a true functional decomposition of the genotype-phenotype map. And we need to keep that in mind. So what do we need in addition? We need some sort of way, of course, to recompose. You see this coming, but we need some sort of, systems level criteria to supplement this sort of decomposition approach here. And of course, the traditional way to do this in systems biology is to do structural analysis of networks. And here I can go really quick because we've been over all of that before. So the traditional way to find modules in networks is to look at the structure of the network. And there are three aspects that are very important here. One idea of this sort of structural analysis is that depending on where a module is in the network, it will have a different sort of function. So you can, in this approach by Davidson and colleagues, you can sort of distinguish between kernels, switches, plugins, and differentiation gene batteries, as we've seen. You can then go on and sort of try to find out what the network does by looking at very fine-grained structure. I told you why this doesn't work before. And we've also been over this, you can look for a criterion of cohesion of modularity, integration, and also distinction from other modules by looking at the density of network interactions. So you look for community structure, for example. So these sort of clusters of interactions are indicative of modules. You see this in a real data set here. And if you're really sophisticated, you can allow for some overlap that represents the pleiotropy of genes, for example, here. Okay, so this is all, and we've been, this is just repetition. So let's just repeat quickly, what is the problem with this? Again, network structure only constrains but does not determine function. So you still don't get a sufficient explanation based on structure alone. Structure does not determine function. I haven't said that in a while, have I? Composition, network behavior can be extremely sensitive to temporal or network context. The addition or removal of a single interaction can completely change the dynamic repertoire of a network. We cannot expect the behavior of a complex network to be a straightforward composite of the behavior of its modular components. All important limitations. And of course, again, static network diagrams don't explain dynamics. Okay, so the thing here is that looking for these structural modules is really not solving the problem. And another thing that you should know about them is it's really hard to find them. It's actually really ambiguous. Often there are problems with the data, but often it's just, they're just not, apparently not there, which is a bit of a mystery. Okay, so structural modules fail to resolve most of the problems we have set out to tackle and create a few new ones in addition. So I'm really not a fan, okay? But again, I mean, use this. It's part of the analysis, right? But it's just not the end of it. Okay, we can use it as evidence and sometimes it works. But the problem is, of course, there is only a loose association between network structure and function. So this doesn't work. Let me illustrate the problem with a beautiful study that was done in James Sharp's group in Barcelona. The first author, Alba Jimenez, did a study that's very similar to this sort of ensemble simulation 2 lectures ago that Cotterell and Sharp did, based on these little circuits that interpreted the morphological gradient. Remember that? So they simulated a huge set of different 3G networks that would interpret a morphogen gradient and make a stripe. So this is a very similar approach. So it's an ensemble approach where you have an even smaller network, 2G network, but now there are some intercellular signaling events going on here. And so you have these networks in neighboring cells, they signal to each other, and they are tasked to do 2 things in 2 different tissues. So that the task here is very different. It's about multifunctionality. So you have one tissue in which there, one tissue here, actually, in which there is a signal here, these purple arrows, which is absent in another tissue. And so you ask the system to do two different things in the two different tissues, depending on this simple difference between the two. There's only a signal that's either there or not. And so in the absence of the signal, the network is supposed to cause lateral induction. So the signaling events in the system are supposed to induce, if a cell is signaling, it's supposed to induce signaling in its neighbor. And what you get are these traveling waves, of course, of signaling through the tissue. The opposite function in the other tissue is lateral inhibition, where signaling causes the neighboring cells to shut off. Okay, and you get this beautiful regular salt and pepper pattern. And of course, this reflects the real world roles that notch signaling plays in development in different contexts. So there's a real sort of specific question here behind this study, but it's an ensemble modeling approach where you just look at all the different possible connections between not just the genes in a cell, but also the intracellular signaling connections. And you sample them, you pick out those networks that can do both of these functions in different contexts. Okay, but the same network, that's important. And so they found something really really interesting. So just like Cotterell, they only found a bunch of different mechanisms that could do both of these things, and they analyzed the circuits, and they found this really, really fascinating result. And here, Zoom doesn't like this, and it crashes every single time I've been trying this. So I'm just going to restart it. I've even tried it outside Zoom, and it worked. So I have no idea what's going on here. It's just Zoom has a dislike for modularity here. Let me share it again. So they looked at the sort of analysis they did is they looked at what kind of nodes and interactions in the network would carry out the two different functions. Okay, and so intuitively you would expect, and I'm just going to show you this like this. Intuitively, you would expect different nodes and different interactions in a network to cause the different functions. So here you have a little example network, and so these three interactions here are causing function A, these interactions are causing function B, and they call this type of circuit a hybrid circuit. So it's just a composite of different circuits. Ideally, you know, that's what you would hope for if you do structural analysis of networks. But on the other extreme, they also found circuits in which all nodes and sort of most of the interactions between them were involved in the different functions. So function A and function B was based on exactly the same nodes. There was 100% overlap in the components that were necessary to implement the two different functions. But most importantly, most of the circuits, most of the mechanisms they found were somewhere in between, where you had a sort of an overlap between nodes. So the functions weren't really cleanly separated. So they called these really integrated circuits, emergent circuits, these really separable circuits, hybrid circuits, but most of the circuits they found were somewhere in between. And so what this means for modular analysis, network analysis, is you cannot expect that there is structural modularity. If you have a network that has no structural modularity at all, you cannot expect either that it's not functionally modular. Okay, so if you just look for structural modularity, you may miss a lot of cases where the network is actually functionally modular. So structural analysis, once again, is not very useful to look at a functional decomposition of the system. Instead, what you need to do is you need to know, just like Jimenez Alba did in her study, what these nodes do. Okay, so we can define a different type of module, which is a dynamical module. And this was first proposed in a paper in 2007 by Dave Irons and Nick Monk, where they write, rather than being a protein complex, a group of co-expressed genes, such a modular, dynamical module can be viewed as the temporal activity. That's very important. Dynamics of a group of genes, proteins. We're going to talk about activity a lot when we talk about homology next time. That controls a specific function in different environmental conditions, cell types, and/or tissues. Okay, to put it slightly differently, dynamical modules consist of a set of network nodes connected by regulatory interactions that implement a particular type of behavior exhibited by the larger network they are embedded in. So modularity is not defined on structure, but on activity here. Okay, and an activity is a certain type of function. More on that later. So let's go and revisit the gap gene system. So we have our models that are fitted to data. We're focusing on Drosophila at the moment. We have our gap gene system once again. And we're looking at this sort of structure of the system. Is there any modularity in this structure? No, it's a very densely connected network. I mean, this is a bad depiction of it because I repeat certain of the domains and the interactions are repeated. But rest assured that there is no structural modularity in this system. And we were puzzled for a long, long time. How could we understand the sort of, the mechanisms for these shifts, for example? I told you ages ago when we looked at the different decompositions of the system, all the different ways in which we tried to take it apart, okay? And one of them, finally, of course, was this sort of, well, there's two ways. Let me explain that first. So one is to sort of look for structural, three ways. One is to look for structural modules, not there. The other one is to look for different behaviors. So we know after our analysis, and I've told you that a couple of times already, that the anterior is very different from the posterior. So you have these sort of switch-like stable boundaries in the anterior and these sort of moving oscillator-based boundaries in the posterior. We've been over that. We've done the phase space analysis. We know there's a bifurcation that's going on in the middle here. Okay, so let's try and separate that out. But then what really actually worked, so that really didn't work either. What really worked was just to look at which genes are active in what region of the embryo. And it turns out that out of the 4 gap genes, only 3 are active in any one place if you go along the embryo's axis. So in the anterior, you have hunchback, giant, and crouple. In the middle, you have hunchback, knurps, and crouple. And in the posterior, of knurps, giant, and crouple that are actually present. And you can do a sensitivity analysis and show that if you eliminate giant from the middle, nothing happens. Because it's not surprising, it's not expressed there. Okay, so simple. And we've also seen this before. So this is one of the different ways in which I showed you we could decompose the system. But the surprising thing here is these sort of decompositions are extremely redundant. Compare them. One, there are always 2 overlapping genes between each one of them. So they're not structural modules. They're not modules in any structural sense at all. And also what's really peculiar here is, of course, they're all AC/DC circuits, right? So we've come back to this simple circuit structure that has a sort of a negative and a positive feedback loop built in. So 3 repressive interactions here between the 3 nodes, and then this double negative, which is a positive feedback loop between Y and Z. So this is our old friend, the AC/DC circuit, and all of those modules are the same circuit. Well, not quite, right? I mean, they have different, just slightly different sets of genes. And therefore, they have slightly different strengths of interactions. And therefore, they will do slightly different things. Okay, now one really puzzling thing was that the bifurcation boundary in the middle doesn't really coincide with the domain of activity of each of these circuits. So it's not that you have one circuit for each sort of activity. Instead, this one in the anterior just switches, okay? It's entirely in the area where you have stable domains switching domains. This one just oscillates, okay? So here, the negative feedback is dominant, and here, the positive feedback is dominant. But here in the middle, you get both, okay? So depending on the maternal concentrations and the initial conditions of this circuit, it either oscillates or it switches. And so it means that it sits right on top of this bifurcation boundary. And depending on the initial conditions, remember from last time, the bifurcation boundary doesn't mean you switch behavior, but depending on where the initial conditions are, you will go to one behavior or the other. So somewhere in here is a bifurcation boundary. So this is both beautifully simple and a little counterintuitive. First of all, the modules aren't really modular in this sort of intuitive sense, structural sense. Okay, they overlap, a big deal. Second, they don't easily correspond with the dynamical behaviors, and yet they explain precisely what the kind of behavior of the system is in each region of the embryo. Okay, so you can really take the system apart based on the activity of these different modules. So what we've done is we've done an analysis in terms of dynamical modularity. Now, why is this important for evolution? And does this, if it's not structurally modular, how can it, lead to modular, discrete sort of traits? And so this is sort of our sort of proof of principle. A simple case study in this case is that you have what is changing. So last time we looked at the fact that you needed to go from the switch-like behavior to the oscillatory behavior to change the extent to which gap domains shift or not. And then between species, we found huge differences in the amount of shifts, right? And so now we can look at these different circuits. They're present in both Drosophila melanogaster and Megazelia, two different fly species, but they behave differently because remember, because of quantitative systems drift, the strength of the interactions in these AC/DC circuits has changed. Okay, so we can now map those changes back from configuration space into the actual network, and we can connect the specific changes in interaction strength in the network to the observed changes in gene expression pattern. So what we find is that this AC/DC circuit here, I already told you, is a multi-stable switch in Drosophila. This one is critical. That's where the critical just means that it's sitting right on top of a bifurcation, and this one is a damped oscillator. And basically, Stuart Kauffman ages ago was talking about the edge of chaos and how living systems have to sit on top of these critical boundaries to be able to evolve. And so this is what's evolvable here. Because what's happened in Megazelia is that AC/DC1 has become critical, while these two posterior circuits have firmly been placed into the damped oscillator region. So this is why you have a different extent of gap domain shifts in Megazelia. Remember, so we had this sort of central hunchback domain, and I showed you a little bit at the end of last lecture that this was the main difference, right? So the extent of the shifts differs between the two different species. And now we have a precise mapping through the process that these different dynamical modules are generating to these differences between the different species. So basically, the spatiotemporal arrangement of gap domains is overall conserved and is very robust to evolutionary change, but the bifurcation boundary between the shifting and the stable regimes is very labile because of underlying criticality. This system is sitting at the edge of chaos, if you want to call it. I find it a bit confusing, the term. It's not really chaos, but it's just a bifurcation, a critical boundary here. And so this explains why certain features of the system are more labile, more evolvable than others. So what we've done here is we've taken this sort of theoretical idea that Irons and Monk had suggested, and we've applied it actually in collaboration with Nick Monk to our real world system. And of course, gap genes are not unique at all. There are such modules everywhere, okay? So they are in different developmental processes, but also physiological processes are, for example, the cell cycle, the systems that control the cell cycle, and so on and so forth. So they're everywhere. So we haven't looked for any of those dynamical modules yet. So we should, right? Because, I mean, they're really sort of dynamical decomposition is a top-down approach, partitioning a network into interlocking subsystems based on decomposing the global behavior of the system, okay? The behavior of the full system corresponds to a straightforward combination of the behavior of its modules. Well, straightforward, but not, it's sometimes a bit complicated, as we've seen for the gap genes here. There is no simple one-to-one relationship between subcircuits and dynamical regimes, okay, necessarily. You shouldn't expect that. And dynamical decomposition allows you to study the dynamical behavior of subcircuits in their native network context. So you eliminate this sort of problem that we had with Uri Alon's network motives that, you know, if you put them together, they don't really behave like they do in isolation anymore. Here you come top down, not bottom up. And this, in this case, is better because you have you study the network in its native context. Dynamical modules are a much better proxy, therefore, for functional modularity than structural modules, since dynamics is much more closely related to function than structure. Okay, so we should be analyzing those modules everywhere, but we're not. And I'm just writing up a paper this summer with Nick Monk about a sort of a new theory of dynamical modularity here, that this is what we should be doing. And see, this is a processual perspective on a genotype-phenotype map. It is trying to go at the architecture of the genotype-phenotype map from the view of the dynamics and activities. We're going to take this topic into the theme, the study of homology the next time. But before we do that, let's look at some of the evolutionary implications of these dynamical modules. Functional modularity of the genotype-phenotype map is a prerequisite for the evolvability of character traits. We know that for a long time. And most regulatory networks are multifunctional. They implement more than just one dynamical behavior. Most multifunctional networks are not structurally modular. So you may be lucky and find structural modularity. But if you don't find it, it doesn't mean that the system isn't functionally modular. Extensive structural overlap between dynamical modules implies that many genes and their interactions contribute to more than one dynamical behavior of multifunctional networks. The decomposition isn't simple, okay? These are complex systems. Even though genes and interactions are shared between modules, their effects on the dynamical behavior of each module are different, and that's important. So the effects will be quasi-independent of each other. The structure of networks and regulatory sequences does not necessarily have to be modular to ensure dissociability of functional modules. That's a central insight here. So if we look at the literature, the prevalence of modular structure in the literature, I mean, this isn't just gene regulatory networks, it's modular enhancers, there's all kinds of things. We find modules everywhere, right? And that's maybe not because they are very prevalent in nature, but because of an ascertainment bias. If we go about finding modules with structural approaches, these are the only modules we're going to find, okay? So we need to go out and look for lots of dynamic modularity here. And this is what the paper that I'm going to write is going to say. So this is one way in which we can move beyond networks in terms of moving beyond structural analysis, okay? Next time, we're going to look at a way of moving beyond networks as moving from character identity networks to sort of the processes that underlie, the activities that underlie homologous processes. So we're going to look at the concept of homology. So some people say Evo-Devo should be studying evolvability. We've done that now. Others say Evo-Devo should be studying homology. We'll do that next time and look at homology from a process, from a truly grounded process perspective. As usual, thanks for listening, and I hope you tune in again next time. Bye now.